And I'm very happy to introduce our next speakers, Coleen Murphy from Princeton. I'll put her slide on.
All right. Thank you to the organizers for inviting me to this meeting. It's been great, and it's been so different from the usual meetings that I go to, and also an opportunity to finally see Copenhagen. All right, so today I'm going to be telling you about work that we've been trying to do to understand how can we extend not really lifespan, but memory span. And as we all know, cognitive performance declines with age. And so what we'd really like to know are there ways that we can search for mechanisms to extend this memory span? How can we slow cognitive decline? So you've already heard today from Colin and Adam about how we can use C. elegans to study aging. Okay, so it's a great organism for finding new mechanisms to extend lifespan. And of course, for us, it's really great because we can do a lot of experiments really fast because it has a really short lifespan. Now, it wouldn't matter at all if evolution hadn't happened, but it did. And so about 70% of the proteins that we find in C. elegans are conserved. So that gives us great hope that what we find in worms could be relevant. Of course, as was already mentioned, one of the things that really broke open this field was this study of a mutant called DAF2, so that's the insulin receptor, and it lives twice as long. And of course, work from Houston and others has shown that centenarians also have variants in this exact same pathway. All right, so how would you take a worm and then try to look for ways to extend memory span in humans or mammals? So can you actually do that? Can you study learning and memory in worms? And if you find something, will it actually be conserved? And even if it is conserved, will it really work? All right, so that was the question that was before us when we started doing this work in memory in worms. Okay, so you probably are wondering, how would you study learning and memory in this little nematode? It's very simple. We developed a Pavlovian learning assay that involves just taking well-fed worms, because they just eat bacteria, and we starve them just for a short time, and then we feed them again while they're smelling a neutral odor. So this is really important, because we want to study memory and learning that is a positive, not just stress-induced kind of memory. And then we ask, when we present them with this odor again, do they choose this, or is it back to a neutral position? And so this neutrality is what we see in the untrained worms. They don't care about the odor. But once we've trained them, they all migrate over to the odor, so that's learning. And if we wait a while, we can look at how it changes with time. So they actually forget this within a few hours. Okay, so that's really short-term memory. And we can study long-term memory simply by repeating, doing space training, as has been done in other organisms. Okay, and then we wait overnight, and then we can test it. And the thing that really makes it long-term memory is the requirement for a transcription factor called CREB. So while CREB is also involved in metabolic functions, it's actually required for long-term memory in every organism that's been studied. And so early on, we did these experiments to test whether it's required. And what we see is that the wild type, this is learning in black, the black bar. This is 16-hour memory in blue. And while CREB mutants, they're fine for learning, they really can't carry out long-term memory. So this shows it's actually basically the same mechanism. We went on to find all the targets of CREB that are turned on during memory. It turns out you can't just compare with the wild type with the mutant. You have to actually go through the whole process of looking at the transcripts that are activated when you do the space training. All right, so that identified a whole bunch of targets. And what was cool for us is that when we looked at what they are, and I realize you can't read these in red, but here's some of these genes. These are basically genes that have been studied to be required in learning and memory in every organism. So if you looked at a mouse paper, for example, a lot of these are cell or neuron papers because of the requirement for their function in mammalian memory. So worms use the same kind of mechanism there. Okay, so I mentioned DAF2, they're long-lived. It turns out they have extended memory as well. And what was curious about this was that they actually use an entirely different set of targets. So what's shown in the big circles are the genes that we found to be up and down regulated in DAF2 mutants that help them live long. But in fact, in these smaller circles, we see those are the genes that are actually regulated in DAF2 mutants in their neurons only. And it's almost an entirely non-overlapping set. So that tells us in the same animal, it's like regulating two different sets of transcriptional targets to do two different jobs, longevity and memory extension. Okay, so that's important to remember because I'm going to tell you something that has nothing to do with longevity. So there's a different process. The other thing that we found was that worms lose their ability to, especially long-term memory, really quickly. So this is a really hard task for them, what we set up. So basically, they lose all these things with time. By the first week of adulthood, they really can't do any learning or memory anymore. But of course, our DAF2 mutants are wonderful, and they extend that time as well with age. Okay, so what we set out to do is ask, It's nice if you can find longevity mechanisms that also affect memory, but can you go the other way? Can you find memory mechanisms regardless, agnostic of their longevity effects? All right, and obviously we want to find things that are conserved with mammals. All right, so we had done a bunch of experiments. We discovered that members of this pathway are required for learning and memory. And so we looked at this the G-alpha protein AGL30. So I apologize for the worm name. It's actually called GNAQ. It's a G-alpha protein in mammals, and it has 82% identity with a G-alpha protein called GNAQ that I'll be talking about. All right, so the question that we asked right away in worms is, if we hyperactivate this gene, will it improve neurotransmitter release or neuropeptide release and have an effect on learning and memory? Okay, so we did that, and we're only training these worms for 1 hour. So that's showing the wild type this is only down here 2 hours. And what you can see is these hyperactivated mutants have greatly extended memory. So we train them for an hour, and they basically, they take like 2 days to forget, okay? And it's the same mechanism that we had already studied because it's Krebs dependent. So it's the same whole pathway. And then when we went on to ask, well, where is it active? It turns out we only require it to function in one neuron. And then when we turn it on in this one neuron, at day 4 of adulthood, when wild-type worms totally have no memory, then we can do the training the next day, and then we test their memory on day 6. It totally rescues their memory. So just the hyperactivation of this gene in one neuron. Okay, so I'm sure you're all saying, well, that's nice. That's in worms. But what about in mammals? All right, so it took me a couple years to convince friends of mine to do this. Just to remind you what I was saying before, they have great similarity to this protein called GNAQ in mice, and this is just our alpha-fold structure here. And so what we did was we asked Saul Vieta in his lab to help us, all right? So the first thing we did was look in what's in published data. So if you look in the mouse hippocampus, you can see that the expression of the mammalian GNAQ it's in the cells that we'd expect it to be functional. So dentae CA1 and CA3, so it's expressed there. And then when we looked at young and old mice, we see that the expression of GNAQ is decreased, okay, in all of those same areas, okay? So it goes down with age. Then we asked a very simple question. Can you just rescue it with more expression, or do you need this hyperactivated form? And we were really surprised by the result. It turns out rescuing with just normal function doesn't do much. You need the hyperactivated form. So we're using the exact same mutation from the worm, now we made it in the mouse, okay? And that's shown here. All right, so that's just these are just tissue cultures. So then we went on to look at what's happening in primary culture, and we see that again, you need this hyperactivated form to get the best result. Okay, so with this in mind, Saul's lab helped us with these experiments. So basically the question was, could we make mice that have the same mutation in it? And can we do it in old animals? Okay, so that's the key here. So we started with 24-month-old mice. This is the beginning. Up until that point, they're just wild-type mice. And they injected and this is Greg Bieri in Saul's lab, injected this hyperactivated form, and then let them recover after the surgery, and then carried out behavioral testing, and then we did more experiments with their brains. And that's just showing the expression there. Okay, so the first thing they did was novel object recognition. So this is a long-term memory task, and it turns out the mice are better at this task. So this is exciting to us right away because this is exactly what we'd hoped. So they do better on this test of long-term memory. And similarly, when they tested the behavior in the radio-arm water maze, the mice also do better. I just want to emphasize again, these are 2-year-old mice. Actually, they're more than 2 years by this time. We started the intervention at 2 years, and this is one of the best results, according to Saul, that he's seen with any of the interventions they've done, even in the younger animals. Okay, so just to remind you, so this is a slide from Saul's lab. They've done lots of work for different interventions, for example, parabiosis and blood factors, and those also depend on Krebs activity. Okay, so what we did then is ask, well, what's going on in the brains of those mice? So they sent us the brains, and we looked for things like, for example, phosphorylated Krebs. That's higher in these animals, as are the expression of immediate early genes. Okay, so things that you'd expect to be activated when you're turning on memory. Okay, so that was good. And then we decided that we'd embark on single nuke-seq so we could actually understand what's going on in the different cell types themselves. And this is actually a fun task because my lab had never done that in anything. And so we found all the cell types we'd expect to find, and then this is differential expression. I just want to note, that we're not seeing new cell types. I just want to mention we don't find things like microglia. We're seeing that basically the same cells are there, but what we see is an increase in lots of synapse formation and organization, structure genes. We see neuron development genes. We didn't expect this, especially axon guidance. So we're seeing things that you normally see in very young mice, now turning on in these old mice, and then signaling cascades. And so what we then looked at was, do the animals actually have more synapses? So again, it's kind of crazy. These really old mice seem to actually have more synapses forming. So this GNAQ seems to have turned on the formation of synapses. Okay, so we have this long list of genes, and usually you'd stop that because it's really nice and you have these go terms. But luckily, we could use the worms to do some fast screening. So we asked, can we go back to the worms and use that as a tool to ask whether the worm homologs of the mouse genes that we found are actually important? I just want to point out, so these are these synapse genes from the mice. When we took the worm orthologs and just did a simple experiment, we just asked, how do those change with age? So everything in red here is a gene that's higher in young animals and declines with age in worms. There's a few exceptions, but the majority are really high in young and go down. And so that's shown here. And these are synapse genes, long-term potentiation, and CREB or LTAM genes, long-term memory genes. And then we use them for behavior experiments. So I have a bunch here highlighted. These are genes that were in our list from the mice, but in the worm homologs, we'd actually years ago already found them as targets and actually had tested them, and they're required for memory. So that's shown here. And then for everything that's not previously tested, we then could use those AGO30 worms that have really great memory, knock down the gene in question, and find out what it does. And so shown here, at least several of these new genes seem to have a role in memory that we hadn't previously anticipated. And so now we have this list. Of course, we could go on and test all those things. But what I want to emphasize is that we've been able to use the worms and the mice together and go back and forth between the systems to really find more targets than we ever would. And instead of saying, well, this is the one gene that's super important, we can actually see how important many of them are at the same time. I will note that this gain-of-function mutation is really critical for seeing these differences. So we see more synaptic gene expression, which, again, I really wasn't expecting to find. A lot of axon guidance genes, so it seems like they're actually trying to grow more, which is surprising. And we see more complex neurites when we do these things in primary neuron culture. We see better function, so they have better memory and behavior, and we see that in both the worms and the mice. But that's not really the longevity stuff you want to hear. It seems that they will work in mammals. And then I want to leave you with one last observation. So Saul, when he had done these experiments, so Greg did the injections, and it was double-blinded. And one day Saul calls me, and he says, I had to unblind myself because I saw something in the cages that I wasn't expecting. And this was it. So they noticed that basically the mice, at least half the mice they were working with, started taking care of their nests again. And so there's actually, I didn't know about this, so I'm sorry if I'm insulting anyone because you know a lot about this, but nestlet, basically taking care of the nest, is actually a metric of health and well-being in the mice. And these GNAQ mice seem to do that. So I can't explain what the mechanism is here because we've only really looked at memory, but it's kind of cool thought that if you improve this one activated protein, you actually might have some potential for improving health span as well. Okay, so we'll be doing more work on that to try to understand that better. All right, so just to wrap up, a few thoughts on lifespan and memory span. I didn't show this before, but you know, these animals, so blue are these Eagle 30 worms, and they don't have better lifespan. They have normal lifespan. So this tells us that there's a complicated connection between longevity and things like memory span or quality of life. You can have one or the other. Certainly DAF2 worms have both. They improve both, but maybe not every intervention that we do will help memory span and vice versa. And so you may not need that for everything. So we should continue to look not only for things that extend longevity, but also things that improve quality of life in the absence of longevity effects, because there may be something valuable that we can find. And if so, then maybe we want to use some additive approaches, things that may help some parts of our body that may, and others that may be specific for our brains. Okay, so that's all the data I want to show. I'm going to thank the people who did all the hard work. So this is a great collaboration with Salveda's lab, particularly Gregor Bieri. And in my lab, Morgan Stevenson led the charge with help from John St. Ange, Rachel Kletsky, Fei Wang, and Xie Zhu. And my daughter reminded me that you guys might be interested in this. Based on a class that I've taught for many years on longevity, I have a book coming out in November. But anyway, back to the people who did all the work. So I will be happy to take any questions.
Thank you so much, Coleen. We have time for...
Thank you very much. Were there any improvement on the motor skills of the mice with the GNAQ?
Yeah, so that's something I'd like to look into more. So when, you know, like Saul and Greg were doing, they were strictly doing all the memory behavior experiments. It wouldn't surprise me if they did based on the other health span, but we haven't tested that yet. It's on our list of to-do.
Okay, I think we will stop here and then remind everybody that we take pictures now outside. I want to thank the speakers of the session that we're actually finishing on time, which is unbelievable. So let's give Coleen another round of applause and the rest.